Hello everyone, I am Qazi Faruddin. Again extend a very warm welcome to all of you for Plant Physiology and Metabolism course. In this lecture, we are going to study the most important aspects of plant physiology, namely how carbon is fixed and what is the path of carbon in photosynthesis. So, just before going into the details, what are the different aspects we are likely to cover, that is Calvin cycle, what are the different reactions of the Calvin cycle, now how carboxylation proceeds and what is the role of Rubisco, and then next stage is reduction, regeneration, and how energy is utilized for synthesizing a molecule of glucose, and then how Calvin cycle is regulated. So, then we will go into the details of evidences for the existence of light and dark reaction. Then what is the source of oxygen in the photosynthesis? Then we will compare light and dark reaction, how these two reactions differ from each other. Now coming to the, if we look this slide picture, we have already discussed that how light is captured by photosynthetic pigment, how it is then transferred to the reaction center, and then electron flow between the two photosystem, that is photosystem second and first, with the consequent synthesis of ATP and NADPH in this process. Now what would be the fate of the synthesized ATP and NADH molecule in the light reaction? So it is more likely that the synthesized ATP and NADPH are definitely going to be utilized in the Calvin cycle. So now question arises where this Calvin cycle is found. So the Calvin-Benson cycle is found in various prokaryotes and all photosynthetic eukaryotes, ranging from primitive algae to advanced angiosperm, and thus serves as a primary pathway for autotrophic carbon dioxide fixation. So it must be clear that Calvin cycle is generally a sequence of chemical transformation converting carbon dioxide and various compounds into glucose. You see in this slide that how carbon dioxide is captured to synthesize a molecule of glucose. Generally what happens in plants, carbon dioxide enters the interiors of the stomatal pores of the leaves, and then as we have already discussed that how it diffuses into the stroma, which is the inner space of the chloroplast. Now this is the site where Calvin cycle takes place. Just a quick recall, what is the site of light reaction? That is in the inner membrane of the thylakoids of the chloroplast, whereas site of C3 cycle is stroma of the chloroplast. So these reactions are also called light independent reactions because they are not directly driven by light. That is why they are light independent reaction. So the Calvin cycle is abbreviated as C3 cycle because carbon atoms from the carbon dioxide are fixed and they are incorporated into the organic molecule and then used to build 3-carbon sugar. That is why it is a C3 cycle. This pathway basically reduces the oxidation state of carbon from its highest level found in carbon dioxide, that is +4, to levels that are characteristic of sugars such as +2 present in the keto group and in secondary alcohols. So due to its remarkable ability to lower the oxidation state of carbon, the Calvin-Benson cycle is also known as reductive pentose phosphate cycle and photosynthetic carbon reduction cycle. So, the complete light independent conversion of carbon dioxide into sugar occurs in basically three distinct stages, namely carboxylation, reduction reaction and ribulose regeneration. So, these are the three different stages of the C3 cycle. So, now we will further go into the details of these three stages. You see, before going into the details of these three stages, let me give you a brief introduction on what really happens in each of them. What happens in carboxylation, what happens in reduction and what happens in regeneration. So, first, carboxylation or fixation of carbon dioxide. In this phase, atmospheric carbon dioxide combines with a 5 carbon acceptor molecule known as ribulose 1,5-bisphosphate to generate two molecules of a 3 carbon intermediate 3-phosphoglycerate. Then coming to the second phase, now what next happen? This 3 carbon 3-phosphoglycerate is reduced to a 3 carbon glyceraldehyde 3-phosphate, or you can say in other word triose phosphates, with the help of energy provided by ATP and NADPH, which were produced during the light reaction of photosynthesis. Now, in the third state, that is regeneration of RUBP, in this phase RUBP is regenerated using additional ATP produced during light reactions. So, the regeneration step ensures a continuous supply of RUBP, allowing the Calvin-Benson cycle to operate repeatedly. Now, we will move into the detailed step of the C3 cycle, that is fixation of carbon dioxide by the acceptor molecule. So, how carbon dioxide is fixed? Basically, carbon dioxide combines with the 5 carbon acceptor molecules just I mentioned, ribulose 1,5-bisphosphate, could be abbreviated as RUBP, and this step is catalyzed by enzyme called Rubisco, that is ribulose 1,5-bisphosphate carboxylase or oxygenase. Here the product formed is a 6 carbon intermediate which is unstable. It is very important to note, this intermediate is unstable. It soon gets, you see in the figure, hydrolyzed into two molecules of a 3 carbon compound called 3-phosphoglyceric acid. The overall reaction therefore combines one molecule of carbon dioxide and one ribulose 1,5-bisphosphate to form two molecules of 3-phosphoglycerate, one of which contains the carbon atom from carbon dioxide, that is reduced form. As the first stable product of the cycle is a 3-phosphoglyceric acid, which is a 3 carbon molecule, hence Calvin cycle is also called as C3 cycle, because the first product formed is a 3 carbon compound. Now, just we will go into the details of what is the key role or pivotal role played by Rubisco. Basically, this Rubisco serves as a pacemaker of the Calvin cycle. So, how Rubisco is important, we will go into the details of interesting fact of the different subunits Rubisco contains, and then we will go into the details how different subunits are encoded by different parts of a cell. So, basically this Rubisco acts as a pacemaker, just I mentioned, pacemaker enzyme in C3 cycle, and it is typically composed of 8 large subunits and 8 small subunits, so a total of 16 subunits forming a hexadimeric structure with a molecular mass of around 550 kilodalton. This is very interesting to note. Large subunit contains the catalytic site for the enzymes, carboxylation and oxygenation activities. It is important to note here that the large subunits are encoded by chloroplast genome, whereas the small units are encoded by nuclear genome. Just I mentioned, it constitutes about 50% of the soluble protein in chloroplast, hence make it as the most abundant chloroplast protein and also most abundant protein in the biosphere. So this fact you should note. Rubisco activity is then regulated by various factors like including the concentration of carbon dioxide, oxygen, magnesium ion and pH. So the pH range of 7.8 to 8 is required for optimum activity of Rubisco. The active site of Rubisco is located in the large subunits where the actual catalysis basically takes place and is inactive in the dark and gets activated in the light. Then how the activity of Rubisco is regulated? Regulation is the carboxylation of amino group of lysine residue that lies at 201 position by carbon dioxide is basically necessary to activate the Rubisco enzyme. However, Rubisco ribulose 1,5 bisphosphate inhibits this carbomylation by binding tightly to the active site and locks the enzyme in the closed conformation as this lysine is not accessible. Now you see that here the role of Rubisco activase comes into play. It basically overcomes this inhibition by causing ATP independent release of ribulose 1,5 bisphosphate. So in this way the lysine amino acid group is now accessible to non-enzymatic carbomylation by carbon dioxide. Magnesium, now next step how Rubisco activity is control. Magnesium is now attracted to negatively charged carbomyl lysine and binds to it, and now the enzyme is thus activated. You see in the figure. Now how 3-phosphoglycerate is reduced. So we will discuss into details of this reduction of 3-phosphoglycerate. That is also very interesting. You see that in the second stage after carboxylation, ATP and NADPH are used to convert 3-phosphoglyceric acid molecule into molecule of a 3-carbon sugar, that is glyceraldehyde 3-phosphate. This stage you see in the figure gets its name because NADPH donates electron or reduces a 3-carbon intermediate to make glyceraldehyde 3-phosphate. So the reduction stage of Calvin cycle, which requires basically ATP and NADPH, converts 3-phosphoglyceric acid from the fixation stage into a 3-carbon sugar. You see in the figure, this process occurs in two major steps. First step is transfer of phosphate from ATP to 3-phosphoglyceraldehyde 3-phosphate. So you see that as you see in the figure, first each molecule of 3-phosphoglyceric acid receives a phosphate group from ATP, turning into a double phosphorylated molecule called 1,3-bisphosphoglycerate and leaving behind ADP as a byproduct. So basically this step is catalyzed by, you see in the figure, by enzyme phosphoglycerate kinase. Now in the second step, reduction of the 1,3-bisphosphoglycerate to glyceraldehyde 3-phosphate. So this 1,3-bisphosphoglycerate molecules are reduced and receive 2 electrons from NADPH and lose one of its phosphate group, turning into a 3-carbon sugar called glyceraldehyde 3-phosphate. So what happens basically, this step produces NADP and phosphate as byproduct and is catalyzed by the enzyme NADPH specific glyceraldehyde 3-phosphate dehydrogenase. Now what happens? Some molecules of glyceraldehyde 3-phosphate are isomerized into dihydroxyacetone phosphate by the enzyme triose phosphate isomerase. The glyceraldehyde 3-phosphate form here basically has many fates in the metabolism. It could be used in starch biosynthesis in chloroplast, or may be transported out of the chloroplast into cytosol to further synthesize sucrose, or it could be used up in the regeneration of RUBP. Also, in some situations, glyceraldehyde 3-phosphate, when converted to dihydroxyacetone phosphate, leaves the chloroplast via a specific transporter, and after reaching the cytosol, it is degraded glycolytically to provide energy and may form what? Fructose 6-phosphate, hence called sucrose. So, the important point to remember here is that the chemical energy of ATP and reducing power of NADPH, both of which are generated using light energy, which keeps the Calvin cycle running. Reciprocally, the Calvin cycle regenerates ADP and NADP, thus providing substrate required by the light reactions. Now, third stage is regeneration of RUBP. The first reaction where CO2 assimilates into triose phosphates consumes RUBP. Now, to maintain a continuous flow of carbon dioxide into carbohydrate, the Rubisco RUBP or RUBP must be constantly regenerated. Now, this regeneration of Rubisco is accomplished by a series of enzymatic reactions. Some of the glyceraldehyde 3-phosphate molecules go to make glucose, while other must be recycled to regenerate Rubisco acceptor. Regeneration basically requires ATP and involves a complex network of reactions. The intermediates in this pathway are 3, 4, 5, 6 and 7 carbon sugars, and now let us discuss each of the reactions in a stepwise manner. So, you see the diagram in the slide which shows the interconversion of triose and pentose phosphates. The number of carbons are represented by black dots. Now, starting with the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which serve as the starting material, step first you see is catalyzed by aldolase, which catalyze the reversible condensation between glyceraldehyde 3-phosphate, that is a 3 carbon sugar, and dihydroxyacetone phosphate to form fructose 1,6 bisphosphate, which is a 6 carbon compound. Now, step 2 you see in the slide, fructose 1,6 bisphosphate is cleaved to fructose 6-phosphate and inorganic phosphate by fructose 1,6 bisphosphatase. This is basically an exergonic and irreversible reaction. Now, we see step 3 is catalyzed by transketolase, which catalyzes the transfer of 2 carbon units of fructose 6-phosphate molecule to glyceraldehyde 3-phosphate to form xylulose 5-phosphate, which is a 5 carbon compound, and tetrose erythrose 4-phosphate, which is a 4 carbon compound. Here the enzyme transketolase contains basically thiamine pyrophosphate, that is abbreviated as TPP, as its prosthetic group and requires magnesium for its proper functioning. Now, you see in the step 4, aldolase acts again and it combines erythrose 4-phosphate with dihydroxyacetone phosphate to form the 7 carbon pseudoheptulose 1,7 bisphosphate. Then in step 5, pseudoheptulose 1,7 bisphosphatase, which is an enzyme unique to plastids, converts the pseudoheptulose 1,7 bisphosphate to pseudoheptulose 7-phosphate, that is a 7 carbon compound. Now in the slide you see step 6, transketolase now basically come into play and acts again and converts pseudoheptulase 7-phosphate and glyceraldehyde 3-phosphate to 2-pentose phosphates, that is ribose 5-phosphate and xylose 5-phosphate. In the step 7 and step 8, the pentose phosphate formed in the step 6, that is ribose 5-phosphate and xylose 5-phosphate, are further converted to ribulose 5-phosphate, that is a 5 carbon compound. Step 9, you see that ribulose 5-phosphate is now finally phosphorylated and converted to ribulose 1,5 bisphosphate. This is catalyzed by ribulose 5-phosphate kinase and ATP, thus completing the cycle. Basically, blue arrow shows the exergonic steps and make the whole process irreversible. These steps are step 2, fructose 1,6 bisphosphate, 5 pseudoheptulose bisphosphatase and 9 ribulose 5-phosphate kinase. Now, what is the economics of expenditure for synthesizing a glucose molecule? For 1 glyceraldehyde 3-phosphate to exit the cycle and go towards glucose synthesis, 3 carbon dioxide molecule must enter the cycle, providing 3 new atoms of the fixed carbon. When 3 carbon dioxide molecules enter the cycle, 6 glyceraldehyde 3-phosphate molecules are basically made. One exits the cycle and used to make glucose, while the other 5 must be recycled to regenerate 3 molecules of RUBP acceptor. So 3 turns of the Kelvin cycle are basically required to make glyceraldehyde 3 molecule, and that can exit the cycle and go towards making glucose. Let's summarize the quantities of Q molecules that enter and exit the Kelvin cycle as 1 net glyceraldehyde 3-phosphate is formed. In 3 turns of the Kelvin cycle, 3 carbon dioxide combines with RUBP acceptors, making 6 molecules of glyceraldehyde 3-phosphate. 1 glyceraldehyde 3-phosphate molecule exits the cycle and goes towards making glucose. 5 glyceraldehyde 3-phosphate molecules are recycled, regenerating 3 RUBP acceptor molecules. 9 ATP are converted to 9 ADP, 6 during the reduction step, you must remember, 3 during the regeneration step. Now, how much NADPH is consumed? 6 NADPH is converted into NADP during the reduction step. Therefore, a glyceraldehyde 3 molecule contains 3 fixed carbon atoms, so it takes 2 glyceraldehyde 3-phosphates to build a 6 carbon glucose molecule. Therefore, it would take 6 turns of the cycle or 6 carbon dioxide, 18 ATP and 12 NADPH to produce 1 molecule of glucose.